Dr. Langer, you don't see us here, but in the room we have Shruti Agrawal, Alan Bash, Robert LaCaze, we have Nick Davis, a lot of folks you have worked with in the past, and we are sorry we're missing you, but clinics come first. And Dr. Borghaei is on the Zoom as well. Shall we get started? Is everybody in a good place? So the initial idea was, when we started off the discussion, was to talk about, we all came out of the IO world. And since the IO world, how things have kind of evolved over time, and how has clinical trials evolved? And also, at the end of the day, though, the core question here is that, given the biotech ecosystem we are in today, how can we help the biotech leaders get to a signal finding fast versus going around and in a wild chase, trying to figure signals out and wasting some of the hard resources that they have at hand? What is it that we can do to help in terms of getting the decisions there? So Dr. Borghaei, if I can start off with you to see how you have seen clinical trials evolving since the glory days of Opdivo and Keytruda and other IO assets. How are you seeing things pivoting? And Dr. Langer, same question as well.
Perhaps it's time to re-look at how to design these trials to try to get to the desired answer without a lot of undue pressure on the cancer centers and on the patients. Your other question regarding how to find a segment is a little bit different, but I'm going to stop here a little bit and ask Dr. Langer to comment, and then we can come back, if you don't mind, to your other question.
Yeah, makes sense. Makes sense.
So I echo what Haas has to say. I think we see the state of Me Too trials, particularly out of East Asia, particularly China, where they're trying to position their checkpoint inhibitor in the fray, and they're virtually identical to the trials we've done here. And it doesn't expand our understanding of how to handle our patients. It's just more drugs to choose from. And to be frank, these drugs are almost indistinguishable in terms of their efficacy or their toxicity profile. If you look at, for instance, here in the U.S., the nivolumab and pembrolizumab, the activity levels are very, very similar. I think we've missed the boat after the initial wave of positive trials, initially second line and then in the front line. And Haas and I both had a major role in all of this, particularly Haas. Things have sort of been fallow. They've been we haven't seen much additional progress. And I think that's in part industry's fault, partly the cooperative groups, but industry's fault for not being imaginative enough. BMS, when they were still interested in 9LA, should have compared that regimen and low PD-L1 and squamous to Keynote 407. They never did. I would have had equipoise putting people on that trial. If it was truly better, we could have proven it. AZ at least has grabbed the reins, and in their Triton trial, they're comparing Poseidon essentially to the more standard Pembro combinations in a population that may or may not benefit more, and they're using biomarkers. They're looking at STK11, HIP1, P53. They're looking specifically at a group, KRAS, at a group that may or may not have a preferential benefit, but it's an ongoing assessment at the next level beyond a current understanding. And I agree with Haas. I think the toxicity patterns of all these drugs are well known. We have to trust the clinicians. Trials that are assessing regimens that have already been published, that are in the New England Journal, to really focus on grade 3, 4, and 5 toxicities and not do the picayune reporting of grade 1 and 2. And we're really not too concerned these days with the duration of toxicities. There is so much paperwork, so much unnecessary detritus that goes with the clinical trial work, and probably 3/4 of it could be eliminated with pragmatic schemes. On the other hand, if you're testing a new drug, you got to go back to basics and do all that.
Makes sense. Makes sense. Try the next question.
It's like, when you look big pharma, and you're facing tough competition, but you have a drug that you really believe is going to have an impact on patient care and advance the knowledge that we have. and hopefully end up helping patients. So how do you get that drug to the clinic a little bit faster? I think to some extent that depends on the kind of drug that you're developing. Obviously, a landscape for a small molecule could be a little bit different than a new biological. It could be a little bit different for a bispecific T-cell engager. It could be different for a CAR T-cell. And that all has to do with the patient population that you're targeting, and also it has to do with the sort of toxicities you would anticipate a particular product and drug and asset to have. Certainly, the first rule is do no harm. And we do understand that in oncology, unfortunately, a lot of drugs that we use does some harm to our patients in terms of adverse events. We haven't been able to folks that don't have those adverse events, but the goal here is to minimize it. There are a lot of innovative phase 1 study designs that have been used over the years to try to get a new drug to an acceptable biologically active dose so that you can not only minimize the number of patients you get an ineffective dose, which ethically you all have a problem with, but also get to a dose level that the company or the manufacturers of the drug, based on the preclinical data, seem to believe that is the right dose for the patient. So these designs have had a huge variability over the years. You can put one patient per dose. You get toxicity. You expand that particular dose. You go one dose level up, dose level down. There are these adaptive designs that have become very popular that we see all the time now in these phase 1/2 studies that we're doing. So again, depending on the target and the drug, there are a variety of ways to sort of move away to some extent from the traditional 3+3 phase 1 clinical trials and sort of build on these more adaptive designs and get to a dose level where you feel a little bit more comfortable. And then opening up to dose expansions. I think there is a fundamental disconnect between what academicians and clinical trialists in academia want and the way the drugs are developed in biotech and big pharma. And that disconnect has to do with the fact that from our point of view, yes, having drugs that we can give to anybody that walks into the door without thinking about it might be a good thing. But now we're in a world where we are actually doing more harm to some of our patients because they truly are not going to benefit from the majority of these drugs. Yet we don't have the tools to select the patients who would absolutely benefit from the drug. And I know I'm asking for a lot. I'm not naive enough to say that, no drug should be developed without a biomarker. But in a way, we need a way to select patients who really are going to either benefit or absolutely not benefit. And that does mean that we need some sort of a biomarker, some sort of an enrichment tool. The more I learn about the biomarker field, the more respect I have for people who do that kind of research because it's very difficult. But at the same time, continuing to develop a drug that we all know is going to work in 30% of the patients and give it to 100% of the patients and then sit back and be surprised when the phase 3 fails because we didn't select the right patient population and we expose patients to unnecessary toxicities is just not a formula that's going to continue to work. And again, we need a shift in our way of thinking and the way we do trials with these targets and with these drugs so that there is an enrichment process. And I think by doing that, we can accelerate drug development and we can benefit a much more focused group of patients than what we're doing right now.
Thank you, Dr. Langer and Dr. Borghaei. Panel, any thoughts or feedback as you hear both our physicians here? Nick, maybe I can ask you, any surprises?
Not really any surprises. I mean, I do think that, and this is a bold statement, but I do think that the way that we run clinical trials today is outmoded. I don't think they're fit for purpose anymore on several dimensions. One is their success rate, and we just alluded to why large phase 3 trials fail. There are many different reasons, but I think it's unacceptable that you see that level of failure. The return on invested capital in R&D is about 4%. So year on year. I think in 1 year, 2021, it was 1.6%. So treasury bonds would be a better investment, right? So there's a reason for that. That's because of the cost of failure. And the most expensive point of failure is a large clinical trial. So we have to get better at that, and I have some ideas about that we can discuss with the panel. I think the second thing is it's enormously inconvenient for the patients. Clinical trials are not designed around patients. So how can we use technology? I know it's not applicable in every therapeutic area, but how do we make the patient engagement in a clinical trial much easier? How do we engage more diverse populations that actually reflect the populations that the drug is intended to treat? Because 86%, I think, of phase 3 clinical trial subjects in the US are middle class and white. And that doesn't represent the population that we wish to treat with almost any drug, I would argue. So how do we increase that? And part of that is the expense involved. Part of it is having to go to a center. So we need to use technology to engage, make it simple for the patient. And I think, one of the other factors is that we don't use the tools, the analytical tools that we have to better design a model and iterate between the economic aspects of a drug, which lots of people focus on, versus how the clinical trial should be designed and powered. What is the minimal kind of viable clinical trial that gets you the endpoint, that gets you the return on investment in the space of the unmet need and gets you pricing and reimbursement? And I don't think there's enough of iteration around that. There's a lot of boilerplate, 80% powered studies, and I think there's ways that we're certainly helping our clients is iteratively model what the minimal kind of viable clinical trial looks like to prove the principle. And that should, I'm not so bothered about accelerating R&D, but I would like to increase the success. I think we can trade one against the other. So, Robert.
Yeah. Shruti, go ahead.
Go, Shruti.
So I wanted to actually expand upon Dr. Borghai's and Nick's thoughts here. The innovation, that traditionally how the drug development is done, there is definitely a disconnect between the academic knowledge, the translational knowledge versus how the drugs are approached, right? And that's maybe driven by the regulatory process also. We collect a lot of biomarker, but in oncology field, we have really struggled to transform that knowledge into the endpoints and into the patient selection, primarily because the hypothesis generation requires a lot of data crunching. And by the time we are designing the phase 3, we have missed that boat. That phase 3 is not a validation. And most bulk of that data is coming out of phase 3, but it's a missed opportunity by that time because the endpoint is not designed that way. The patient population is not designed that way. So maybe the innovation is needed about how do we develop the diagnostics and the biomarkers, and not necessarily just one single biomarker, maybe the composite biomarkers. That's where we can actually identify the patient population and some stat as well as the regulatory innovation to kind of make sure that that is incorporated in appropriate patient treatment and the label.
Robert. No, I was going to say a very similar thought, that if you're having to, do EKGs and bringing patients back in every 46 weeks on a phase 3 trial, probably miss the opportunity in a phase 2 translational work to really identify the patient population, and you pay for it in your phase 3 because now you have to collect the data to understand really if your trials fell, why did it fail? Or what patient population actually benefited versus the ones that did not. And so I think, you start with the end goal in mind, even in your phase 1, as you're at least in the oncology space, as you think about the work that needs to be done, the signal finding, the translational, the biology, making sure that, again, when you have two drugs that are well known and you're putting them together and you're looking at, I think that's nothing's easy, but that's easier. And I don't know why one would want a lot of data collection beyond just, the what you would need from a regulatory approval standpoint. But I think there's opportunities in the phase 1 and phase 2 to really get the translational medicine work right. And that becomes a very important trade-off issue from a biotech standpoint. Now, large pharma obviously have the resources and they can actually do that. But on the biotech side, maybe, these extra 2 or 3 pieces of data you need to collect is going to either hold you up another 4 or 5 months in a phase 2, or it's going to add costs that you can't afford. So, these are very important trade-offs, but when you get to your phase 3, if that work hasn't been done, it's going to cost a lot more in the long run. So those are just all kinds of considerations that have to be taken into account as a small biotech company versus, say, a large pharma company.
Alan, did you?
Good.
I think so. Somewhere between an overly complex clinical study and the minimal viable is probably where we're going to land on a number of these, because at the end of the day, there are regulatory requirements. And then the pharma company comes along and says, we really want to look at this because this is going to add to the evidence package for access. And then someone else comes along and says, well, we really need to understand this from a translational standpoint because it's an interesting research question. So, I think partnering with, Dr. Langer, Dr. Borghaei, and their colleagues, figuring out exactly what is not needed is a very good goal. But again, on a new molecular entity, you're going to need, it's the FDA that's going to mostly dictate how many times you scan a patient, or how frequently you're testing the adverse events. I think it's probably just somewhere in the middle we have to get to.
I want to give some chance to the audience here if you have any questions for Dr. Borghaei or Dr. Langer. Anyone before we go back to the panel here?
So traditionally, the R&D thinks, we all think the phase 1 first in human trial is really for safety and dose finding. And now within oncology, we're adding efficacy exploratory, go into that study. Now we're talking about biomarker translational. So I wanted to understand the panel's thinking about what are how is that the general expectation of phase 1 first in human trials? Where is the balance we should, considering the speed versus the quantity and quality of the data for phase 1?
Maybe we could let the two clinicians give a thought.
Yeah, that's what I was saying. Dr. Borghaei, could you hear the question?
I think, and most of it. So the question really is, do we need to change phase 1 design, and how do you do an appropriate phase 1 before you move on? Is that basically it?
Yes.
So I think I'm a little too old to get very innovative. I don't know. I kind of still like my phase 1s to be a phase 1. I kind of want my phase 2s to be phase 2s. But, what I see, obviously, is that I agree that phase 1 is usually those finding safety. And I don't see any design that's going to get away from that, at least for the time being. And I don't know if the regulatory environment is ready for something that robustly different than what we're doing. Maybe we can get away from the 3+3 design that we've been using for an eternity. But I think the phase 1s have their own sort of life and development, and it is what it is. What we're seeing are these multi-cohort phase 2 studies. And depending, again, on, I'm not saying everybody does this, but what's become very popular is that you do a rapid dose escalation, you define some sort of an MTD. And by the way, we still are doing MTD, meaning we're going to make people sick intentionally, and then we're going to come back. And again, that hasn't changed for forever. And I don't know if that's a viable option going forward. And then we're not considering maximum biological dose. Again, we're not using technology, as somebody on the panel suggested, to try to figure out, we really have to make people have, great for nausea before we say, okay, this is too much, let's back down. So anyway, we define the dose, and then we open multiple cohorts. And all of a sudden, the phase 1 that used to be 30, 50 patients, now all of a sudden is a phase 1/2, and you have like 400 patients. And the issues here are that when you open these multiple cohorts, very few of these studies have any kind of futility stopping rules, meaning, we want to put 40 patients on and just see what happens. Well, I don't think as a comprehensive cancer center, it's a job to put 40 patients on a study drug that has minimal activity or toxicity data associated with it just to see what happens. I need something that tells me if I treat the first 15 and nothing happens, I'm going to stop. I'm not going to continue with that cohort. We're not getting that. I want to see a study design that says out of the first 15, if 20 of them end up with grade 4 toxicities and 3 of them are admitted to the hospital, I'm going to hold and not put patients on that cohort. But we're not seeing that. So there's this emphasis on rapidly putting patients on studies without any hesitation, without any checks and balances that we normally want in it, because we're in a competitive environment and we want our drug to finish this phase 1, 2, and then jump into a phase 3. And I think one of the other reasons for phase 3s failing is because this rush to be able to do all of these things in a very rapid procession in one protocol, regardless of the burden that it puts on the patients and the clinical trial officers and all the cancer centers and doctors who are supposed to do this. So again, I know I'm sounding like I'm complaining, and I'm not. We do this all the time. I'm just highlighting some of the difficulties I see from an investigator side, and the side that perhaps some of you on the panel don't see is it is me who walks into the patient's room, or is Dr. Langer walking into the patient's room, trying to manage toxicities, trying to explain what happens, and trying to explain what the next steps are. So some of these issues with the way we're doing trials right now are becoming a little bit unmanageable for cancer centers and for clinical trials offices. So we definitely are going to be pushing back, and we're probably not going to participate in as many studies just because we have to deal with a lot of these other issues as a side. But the solution to it, again, in my view, is to try to select the dose a little bit more carefully, define the toxicities a little bit better, try to see if there is a biologically active dose as opposed to the most toxic dose of the drug, and then do very well thought, phase 2 studies where we all can learn something from it with the appropriate futility and toxicity stoppings, just so that we don't end up, unnecessarily putting patients on these studies. And it might take a little bit longer, but I think it might actually help us do better phase 3 studies. Again, I will never be enough to say that if you do anything like that, your phase 3s are going to succeed. But I can tell you confidently that if you do an appropriate phase 2 study, you might decide not to proceed to phase 3 with a specific asset. Right. And I think that's going to be the advantage.
Dr. Langer, yeah, go ahead, please. We wanted to get your thoughts as well.
Continuous reassessment methodology is something that we haven't adequately exploited. You have 1 or 2 people at an early dose level, no toxicity. You don't wait till 3 to 6, you start escalating. But then you continue to accrue at that lower dose level as sort of an expansion cohort. And that way you don't go through these fits of stops and starts. The hardest thing for us as clinicians, having a clinical trial open and being told, oh, it'll be another 4 weeks, another 6 weeks, another 8 weeks so we can enroll your patient with cancer doesn't take a holiday. So we really need access to these, and we certainly gather importantly viable data on these patients as we accrue. So much more flexibility in clinical phase 1 trial design is absolutely essential, and hasn't really been adequately exploited. Basically, any new drug can be approached this way, particularly if they're non-toxic or relatively non-toxic. I think we missed a big opportunity in phase 2, particularly in randomized phase 2, for doing appropriate exploration of biomarkers. And a case in point would be potentially datopotamab deruxtecan, the ADC, which went very quickly to phase 3 based on reasonable data in phase 1 and early phase 2, but without a biomarker, or at least without an established biomarker. Everyone, virtually all patients have TROP expression. They sort of backed into the biomarker, but only after the phase 3 was completed. This NMR ratio, the nuclear cytoplasmic membrane ratio of TROP expression, they were finally able to identify a cohort that may truly have preferential benefit. To be honest, I think they're going to have to redo the phase 3, and it would have been so much nicer if that could have been done as part of a randomized phase 2, or at least gotten the data secured that way. And then we have object lessons of what absolutely should not be repeated, and that's the tiragolumab experience in small cell, excessive stage small cell. There wasn't a lick of preclinical data. There was no early clinical data. Knowing the drug was relatively non-toxic, they went right ahead and compared standard chemo, in this case IO, the checkpoint inhibitors become part of the landscape, plus or minus durvalumab, and didn't see a lick of benefit. Major expenditures, expenditure of time and money and labor, with really no foundational support, a priority before going ahead with that sort of trial. As one of my colleagues said, well, at least we didn't hurt anybody, but that's a very bad excuse for going ahead with this phase 3 that doesn't have the adequate justification beforehand. So the pragmatic trials really can only apply once the drugs are approved. I agree very much that in the early development process and in the process of getting an FDA approval, that sort of approach is probably not the easiest. But once the drug's approved, once we know that it's potentially safe, and in combination with other agents, we can probably bypass a lot of the unnecessary work that seems to be inherent in all clinical trials. Good.
Yeah, I want to say that we also other component that we're not representing on the panel today, and that's the FDA, of course, that has a lot to say about the way we design drugs and not to get them and all of that. So I think that is something that I think one of the panelists, unfortunately I couldn't quite hear, did bring up, but the regulatory environment also needs some attention and discussion, obviously.
Yeah, I know Dr. Langer and Dr. Borghaei has to leave in 5 minutes. Maybe I can. Do you guys have any other questions for the clinicians? If not, I met Dr. Borghaei in Spain over the summer, and over dinner he goes, now I'm throwing you under the bus. It's like, you're such a copycat industry, right? So how can we avoid, it's true. And I think Dr. Langer kind of said the same thing. I remember meeting Dr. Langer the first time, and he said the company was having 4,900 patients on 3 different lung studies, totally undifferentiated, didn't even know what they were going to study and why they were. And we never knew what happened to those studies, right? Thank you, Dr. Langer, Dr. Borghaei. I know you have patients waiting, and thank you for taking time out of your clinic days to see us. Next spring, we'll do it again, but we will do it when you don't have clinic days. So we'll continue with the panel here. Thank you for joining us. All right. So with the panel here in the room here, we will go in the chronology of IO then. And you're the TIGIT lady over here. What to make of TIGIT? There are multiple different variations of narrative. What do you make of it?
So I guess for TIGIT story to unfold, we can also look into the combination IO story with either the anti-PD-1, anti-CTLA-4, or anti-PD-1 LAG-3, right? So the TIGIT data right now in public domain is very limited. We have like either gastric cancer or lung cancer data, but we don't know what is the driver of the efficacy, whether it's limited to particular segment of anti-PD-L1. All that needs to be unfolded. Yes, there had been some negative news about TIGIT, but we don't know how that data is being interpreted. So we are waiting to get into the data. However, there are few anti-TIGITs who are differentiated, including the bispecific as well. So what's the role of that? Whether we would be able to actually develop TIGIT similar to anti-CTLA-4 or anti-LAG-3 would be, like, time would tell, and there are trials going on. So I do hope that there is a role of anti-TIGIT, which we know that depending on the safety profile or depending on the toxicity profile of anti-TIGIT would kind of be able to develop it accordingly. So waiting for data for the differentiated anti-TIGITs and bispecifics.
Absolutely. We're hoping for good results as well. It's good news for patients. Alan, so you come from the area of, you don't have to be stuck in ADC land either, because you have done other things. Beyond checkpoint inhibitors and others in ADC, going back to our Opdivo days, what are some of your thoughts in where clinical trial is going, and how can this segment pay better attention?
I liked what Dr. Borghaei and Dr. Langer talked about. You can hear their frustration, and what's great is, we have to hear that perspective, because we come at these with very genuine intentions to do the right thing for our companies, whether it's a small company or a big company, and that you see like these well-intended decisions just completely drive the investigators crazy. The need to stop and start in phase 1, the lack of flexibility, the lack of stopping rules, the lack of futility. And we talked about phase 2, the biomarker, and not wanting to make a decision on the biomarker too early, but they really want to know that because that's going to help inform the phase 3. Specifically with ADC, I'll just share a little bit about kind of how the ADC landscape is playing out. I think you showed one of the graphs, John, earlier about the ADC space. So, there was tremendous excitement with HER2. And then Dr. Borghaei talked a lot about TROP2, and you heard from him. So then everybody rushed in, and we do suffer a lot from Me Too. So there's HER2, TROP2, and Me Too. Those are the 3 targets we always hear a lot about. And what happens is if you go, like I've gone to several of the conferences where a lot of the ADC technology is being touted. If you have so the 3 components obviously of an ADC are target, linker, and payload, right? And if you have a novel linker, for example, let's just say that's your core capability as a small company. You've got a new linker. You do not want to play around with a novel target. You don't want to add the novelty on top of novelty. So what do? You do a HER2, good old-fashioned HER2, and you change the linker. So that's why some of these decisions get made where you start to have everyone chasing the same targets. Zeal Bio was actually looking at the opposite, and we were looking at the fact that, yes, the lion's share of all ADC trials are circling around like 9 targets. So we talked about TROP2, HER2, Claudin 18 is another one. We were looking at a novel target. Because of that, we actually wanted to use very non-proprietary or sort of good old-fashioned off-the-shelf linker and payloads because you don't want to add too much complexity. So depending on where you're sitting, depending on what decision you're trying to make and what value you're trying to add, it might inform some of these decisions. I do think we certainly suffer from this herd mentality. I also, you see that now with GLPs, right? I mean, it just plays out in every field. And the other thing is, I'll just make one other comment. Sorry, I know I'm going on, but the pharma companies, the big pharma companies, there's this whole risk aversion, right? You have a drug, it gets approved. Now you want to expand the usage to other indications. Do you want to start playing around with the dose at that point? No, right? Because that might have impact on pricing. It might have impact on implications on the original indication. So you don't want to touch the dose. You don't really want to start sub-segmenting your population at that point. You should have done it probably 10 years earlier. And you don't want to try to go up against another standard of therapy because you might lose and then the whole thing falls apart. So, we're a victim of our own success. We get an approval, we get a drug, we want to expand, but then we're very risk averse to try to answer some of these unanswered questions. Make sense? Robert?
Oh, also, I did want to ask you, you come from obviously all of the other backgrounds we're coming from, but then you have the radiopharma on top of that. Any nuanced approaches from that segment?
Yeah, so radiopharma is, to me, is a very exciting up-and-coming field. Even when I started in this industry, the saying was you either cut it out, you burn it out, or you poison it. And that was really the 3 ways of treating cancer: chemotherapy, radiation therapy, or surgery, or a combination of the 3. Obviously, we've come a long ways since those days. But now being able to harness the radiopharmaceutical approach, I've been able to go beyond just doing targeted radiation, I think is very, very exciting. Now, it's also very tricky because, with that approach, when we look at the data in prostate, it's pretty exciting what's happening in prostate cancer with the radiopharmaceuticals. The question is, can you go beyond prostate cancer? Right now, the two drugs, one's a beta and one's an alpha, and the alpha is not linked. It's a naked radioisotope, and both have had overall survival advantages. The question then becomes, all right, who's going to win in this space? Is it going to be long-term? Is it going to be the alpha? Or is it going to be the beta? Is it going to be the copper? Is it going to be the lead? So there's all kind of different isotopes that can be utilized out there. I think the second key point when you think about the radiopharmaceutical space is doing a phase 1, you really need to go to clinics that do radiopharmaceuticals because they're so different than small molecules or biologics. And you have to have people who really understand, how to manage and treat the patients from a radiopharmaceutical standpoint, because like cell therapy to some degree, you have to have that whole manufacturing component that has to be built into it, because each drug, once you make the drug, you have maybe 7 to 10 days before the drug is below specs because of the decay from the isotope, which obviously can impact your phase 1 study as well. So there's so many different components to this, and but if you think about it, and your point about not having two unknowns when you're doing drug development. If you're using technology, ADC technology, and you're trying to target a new target, but you put the isotope, for example, on the ADC, because you're using such a different warhead, if you will, you can still go after many of the same tumors and the same targets, because you're using a very different modality. And which makes it exciting. And then we all know that when you look at some of the early data of radiation in combination with the PD-1 inhibitors, the checkpoint inhibitors, there seem to be some synergies there, and it would make sense that there would be. So the other question then is, how do you put the radiopharmaceuticals in with the IO type of drugs? And it's a stepwise approach. So it's got which radioisotope is going to win, and then which tumor do you go after prostate cancer? What type of ADC do you want to utilize? And then eventually, how do you combine with IO in all of those tumors where IO is already there? And do you combine with chemotherapy and the other tumor types? So this is why I think the radiopharmaceutical space can be the next wave of explosive growth, but it's still very, very early on in the stage. And one of the biggest issues in the radiopharmaceutical industry has been just supply. Just being able to get the isotopes themselves, especially when you look at some of the alpha drugs, really, really difficult supply has been a challenge. And so, thinking about back to the clinical trial piece, what sites do you go to? What CDMO, what manufacturing organization do you want to partner with, and how do you get that supply chain from your and it needs to be scalable at some point in time, assuming you're going to be successful. You want to be able to scale that as quickly as possible. And so there's so many things that go into the radiopharmaceutical space, but I find it to potentially be very exciting.
Thanks for the input. Nick, do you want to round this up?
I'm not sure I can add much from my learner trends here, but just in general principles of the amount of budget distribution between research and development, particularly in large pharma companies, is about 20-80. So 80% of all the R&D budget is spent in the clinical space. And if you think about the things that we're putting into the clinic, that there's no biomarkers. We can't select patients. We don't always have a very well-validated target. It's not always possible, but I often think it should be somewhat, if not inverted, at least let's spend more time validating novel mechanisms before we actually even think about going into the clinic. I just think we rush, as my point earlier about I would take more time. I'm fine with that, but I'd like to see an increase in success. And I think just thinking outside of oncology clinical trials is that it's increasingly difficult to recruit subjects into clinical trials, not necessarily in oncology, but in almost every other therapeutic area, because the Gen Zs of the world and slightly younger, or slightly older than that, but certainly younger than me, you want everything instantly. Like if you can't get something from Amazon within 3 hours, then you don't buy it. So how are we going to engage that generation in clinical trials? Because if we don't have any clinical trial subjects, we don't have any new drugs. So anyway. Just a general point.
Thank you, panel. The discussion continues in a little while with Giovanni leading into more honing into the trial designs, et cetera, as well. But thank you for taking the time now.
Thank you.
Thank you.